So I want to talk about case study because a few people get a little bit confused by it. So case study is all about the delaying of gratification. So can people resist the temptation of something which is quite attractive in order to achieve some kind of goal? So the theory behind this, the ability to resist temptation in favour of long-term goals is an essential component of individual, societal and economic success. So it's successful for the individual, it's successful for society, and it's successful in terms of achieving money. You know, if you think about it, in order to delay gratification on an individual level, you might decide that instead of going to a party, you're going to revise for an exam, and then on that exam you do really well. So that ability to delay that gratification has meant that you become more successful. On a societal level, people will, study more and achieve degrees and things like that, and then get jobs in that sense, and so they become successful and contribute to society in a more kind of, contributal kind of way. Because they're delaying the gratification, they're not just going for instant gratification of something, they're thinking about in the long term what impact that's going to have, and that's better for society. And it also means that that individual is going to, be successful economically, and also that the country will be economically successful also, because everybody within it is working towards long term, thought out, well considered goals, rather than just that initial temptation to something that could spoil that. So alluring situations can diminish control. So alluring kind of means just like attractive, something that is, something that you want basically. And this alluring situation means that you've got to have a capacity to control your impulses. So, you might want something, but your brain will help you control your desire for what it is that you want. And this obviously changes with age. Children are going to find it very difficult to resist the temptation of something like a sweet, if that sweet is something that's alluring to them. Whereas as they get older, it's not going to be as difficult to do that. So it's changing with age. Now, delay of gratification depends importantly on cognitive control. So you might want this kind of reward straight away, but it's your cognitive control, how you're thinking about something, that's going to decide whether or not you will resist the temptation and delay gratification, or whether you would just want that immediate gratification, so want the reward straight away. And individuals tend to use kind of different cognitive strategies, so this kind of basically means different ways of thinking in order to allow them to delay gratification. So they might try and distract themselves, try and look away from the object. Say it is a marshmallow, the child might try and look away from it. There's a video of children doing the marshmallow task, and you can see lots of them trying not to eat it by maybe smelling it, or like kind of pushing the plate away from them, that kind of thing. So they're, strategies of cognitive control. As you get older, they're obviously going to get a lot more sophisticated. So you might come up with something else, so that you don't want that immediate reward, and you can think of long term what the benefit is going to be of delaying that gratification. So this is the video I was talking about. If you watch this, you can see kind of how the children are behaving when they're given the choice. This little boy here is sniffing it, as I mentioned before. So the theory in terms of the brain structures, the brain regions, we're looking at the biological approach with the study, to use it as a support for the biological explanation of behaviour. Jabi and Cases in 2008 demonstrated that the inferior frontal gyrus area of the brain, as demonstrated on these two diagrams, is involved in our interpretation of facial expressions and the corresponding emotional response to those facial expressions. So, if you see a face that is quite angry looking, your emotional response to that is to be quite scared, because that person might be aggressive towards you. If you see a face that is smiling, then your emotional response to that that's appropriate would be perhaps to smile back, because that person is not showing a threat to you. So the inferior frontal gyrus is, involved in that, so that it's kind of almost like a controller, a kind of, a mechanism to help us respond in an appropriate way. A correlation has also been found between the the avoidance of risky behaviour, and a greater excitation in the right inferior frontal gyrus. So when you're trying to avoid risky behaviour, there's more activity in that right inferior frontal gyrus. So again, it's kind of almost commanding to avoid being a risk, have that correct emotional response to something that's being seen. As I mentioned before, if someone's angry, avoid that risky behaviour, and retreat or something. That'd be an appropriate avoidance of that risky behaviour. It's been found that functionally, the ventral striatum facilitates and balances motivation. So it kind of is linked to what do you want to do in terms of what you're motivated to do. And it says it's with both higher level and lower functions, such as inhibiting one's behaviour in a complex social interaction. So, perhaps somebody says something and you're quite angry about that, but instead of being aggressive towards the person, your ventral striatum wants to be aggressive towards the person. You might then inhibit that because it's not appropriate to be like that. Say, for example, you're at work and you're angry at your boss. You might want to be angry towards your boss, but it's not appropriate in that situation to behave like that. So your ventral striatum might balance that motivation so that it inhibits that behaviour of being an appropriate aggressive response. The region that's been found to be I was going to say that, I just read that completely wrong. This region has been found to be the region in the basal ganglia neural circuit, mostly associated with reward. So you've got the ventral striatum in the basal ganglia neural circuit. What that means is it's within kind of a collection of structures. The basal ganglia is all about movement and reward. So this is kind of like where it's linked to the motivation. If something is quite rewarding, the ventral striatum is going to be motivated towards achieving that. So basically, the ventral striatum is wanting that kind of reward straight away. It kind of wants immediate gratification. So brain imaging studies, often with kind of a functional MRI scan. So these kind of functional MRI, because you're seeing bits light up on them. If it was just kind of like a black and white film, that's just an MRI scan. So this is a functional MRI. The bits that are kind of orangey coloured are the bits that have got the most activity. The red's obviously got activity as well, but the brightest colours are usually the ones that are the most activity. On some scans, it's if it's blue, green, yellow and red, the red shows the most activities. It depends on the scan. But on this one, it's provided evidence for separate brain systems that are related to immediate gratification and long-term choice behaviour, so that delayed gratification. So we have different areas involved in that. And on this scan here, it's showing the ventral striatum is being activated during those go and no-go tasks. Imaging's also shown that a region of the prefrontal cortex, the inferior frontal gyrus, is involved in resolving interference between the go and no-go and the cognitive tasks. So basically, the ventral striatum wants the immediate reward. It wants the immediate gratification. That's the bit where you get the reward sense of the brain, so it's got motivation to seek that reward. It's quite hedonistic in that sense. But the inferior frontal gyrus is the one who's kind of cognitively kind of thinking about it and going, okay, no, that's not appropriate in this situation, so I will delay that gratification because I want a long-term reward. So that's kind of the one that's a bit more what you're kind of thinking of, more of a conscious. Ventral striatum is more of kind of like the primitive. It wants the reward straight away. So you can remember these by thinking of the ventral striatum as Thomas Tank Engine. Thomas Tank Engine loves train. No, he's a train. Wait, he loves riding on the tracks. So he gets enjoyment out of that, and he wants to do it all day and all night. But he can't do it at night time because he'll disturb everybody in the village where he lives. So the fat controller is like, no, you can only go when I say you can go, and also you can't go whenever you want because you might crash into all the other trains. So the inferior frontal gyrus is the one that's deciding when you get the reward. So do you get it immediately? Do you get it? Are you delaying it? They're the one that decides when it's going to happen. Whereas the ventral striatum wants it immediately. Now, this is a good example of the go and don't go tasks, or go no go tasks, as they're also called. It's kind of to show, that decision of should you go, should you not go. And what that means is, like, they tell you to press a button. So it might show you a picture of a face, and they say, okay, you're going to press the button every time it's a female face. If a female face comes up, that's a go. If a male face comes up, that's a no go. So it's testing whether or not people will go or no go, and if there's any, opportunities where that changes. So they've got here go and no go tasks, different ones showing. So these are just examples of them. Previous research has showed that performance on delayed gratification tasks in childhood predicts the same efficiency that an adult will have on these go and no go tasks. So it's suggesting that this characteristic of being able to delay gratification is continuous over time. The people who, as preschoolers, direct their attention towards rewarding aspects of the task, such as focusing on the cookies or the marshmallow, whatever they'd use, so the high temptation focus group, they had more difficulty suppressing those inappropriate actions than the low temptation focus counterparts. So what that means is the children who kind of had those strategies to look away and to think of other things were more likely to delay the gratification, whereas the children that were sniffing it and kind of like focusing on it and thinking about how amazingly moreish and tasty it would be, they were less likely to be able to suppress these inappropriate actions, and they wanted immediate gratification, and as this child is doing here, would eat the cookie. So findings suggest that performance in preschool delayed gratification may predict the capacity in adulthood to be able to control your thoughts and try and stop interference from other information. So there's lots of different studies that have been found to support this. And they've also found that alluring or social context can diminish self-control. So all these faces here are considered to be alluring because they're all kind of like very happy or very kind of like angry. They're kind of alluring in the sense that we need to respond to those. Evolutionary speaking, if you were to see these faces, you'd have to have an appropriate response in order to survive. So that's kind of where our brains have kind of got this from. This is why it's faster. We can process it faster because we could be in danger if people are showing certain faces. Baring of the teeth, even though it's a smile, baring of the teeth signals danger. So we process that kind of information faster. Whereas if these faces were neutral, it would be slower. So we'd be able to kind of not have as much interference from it and be able to control our kind of impulses from our ventral striatum. So the aim of this study of cases was to build on all this previous research that's been done and to assess whether this delay of gratification in childhood does actually predict the impulse control abilities and the sensitivity to alluring or social cues. So for example, happy faces. At the behavioural levels, that's how the person is actually behaving. Will they eat the marshmallow? If it's the marshmallow test, will they complete the go no go test with accuracy or not? And then also at the neural level, so they've got the scans of the brain. Is it actually showing differences in those different brain regions as well? So the ventral striatum and the inferior frontal gyrus in terms of when they're adults. So, does the childhood result predict what they're going to be like in adult? These alluring qualities of targets in an impulse control test were manipulated to be able to examine these behavioural and neural correlates. Neural correlates just means whereabouts they are in. So correlate, where does it correlate? So neurally, whereabouts in the brain is it? So what structures is it in? That kind of thing. And they're basically using a functional MRI scan. That's what I mentioned before. It looks like this machine here, and you get scans on it. The scans that you can see there are just stills from a computer, but on a computer they'll show kind of glowing images of where things are being activated and stuff like that. So images can be manipulated to show that at the neural level. So in terms of the methodology, how it was done, it's considered a quasi-experiment because you've got a naturally occurring IV. It's whether the child is a high delayer or a low delayer. So basically, if the child could With the marshmallow task, they've been told that you've got one marshmallow in front of you. If you eat the marshmallow straight away, that's fine. But if the marshmallow's still there when the researcher returns, you get another one. So if they eat the marshmallow straight away, or they eat the marshmallow before the researcher comes back and offers them the other marshmallow, then they're a low delayer. They can't delay that gratification very long. It's a low delayer. Whereas if the child can wait until the researcher comes back and has another marshmallow, they're a high delayer. They can delay that gratification to a higher level because they can wait for that second marshmallow. The dependent variable what's being measured is the performance on the impulse control test, the go no go test, so in terms of reaction times, how fast they are and how accurate they are at that. So that's in experiment 1. And then also the performance on the impulse control tasks in terms of reaction times and accuracy, and the imaging results from the fMRI scanner. So looking at the behavioural level in experiment 1 and then experiment 2 looking at the neural correlates. As the participants completed self-control scales, so this is kind of like on a self-report measure, so they're kind of rating themselves and whether they think they can control themselves, essentially. And they did this when they were in the 20s, and they also did it when they were in the 30s. And they did experiment 1. They did both the hot and cool go and no go tasks, which the hot one would be seeing the happy or the sad faces, the ones that are quite alluring, as we mentioned before, that are quite fast to process, and the cool ones, which are the neutral faces. Because they were taking part in so many things, and they're doing it a couple of times, so in the 20s and in the 30s, it's a class of repeated measures design. They're taking part in quite a few tasks, although they are only taking part in one condition of the IV. They're either a high delayer or a low delayer. So it could be classed an independent measure design for that reason. So it gets a bit complicated there. But this is what the exam board have given us in terms of saying it's repeated measure design. So perhaps just go with that one as long as you explain why. It's also a longitudinal study because you're following some of the original participants from the age of 4 years until they're in their 40s. So it's looking at how their behaviour is changing over time. And looking at changing behaviour over time means that you're going to get a lot of qualitative data, or if not qualitative data, a lot of quantitative data to analyse and compare participants with. So it suggests that the findings are perhaps more valid for that reason. They're more accurate at suggesting whether people can delay gratification, whether it's consistent across ages. Now, the sample starts off with 562 children, 4-year-old pupils from Stanford Bing Nursery School. So Stanford University is a famous university, does a lot of research in various fields, but in psychology there's a lot of studies that have been done at Stanford, and they've got a nursery school attached to it. And they're completing this delayed gratification task in the late 1960s and early 1970s. It's quite a big sample, but they've done it over 10 years. Well, not 10 years because it's late 60s and early 70s, so probably a couple of years, to see whether or not these children can delay gratification. And 155 of them completed the self-control scale in their 20s in 1993, and then 135 of them when they were in the 30s in 2003. So really, this sample is suffering from a lot of attrition. A lot of participants are dropping out, and this is perhaps because they just don't want to take part. They've perhaps moved location, or the researchers haven't managed to keep in contact with them. They've not replied to letters, or however they've been tried to be recruited, well, they've been recruited but contacted afterwards. So lots of things could happen, which means that people just drop out of the study. So we've got them dropping out. We saw quite a large sample of 135 in the 30s in 2003. Now 117 of the 135 individuals that are left, who were above average or below average in their original delayed gratification performance, so on the marshmallow task, if they were above average or they were below average in terms of being above average for a high delayer or below average for a high delayer and for a low delayer also, as well as on the self-report measures of the self-control, they were contacted to take part in the actual procedure of this study. So 59 of the 117 agreed. So again, we've got quite a low response rate to this. we've ended up with quite a small sample of 53, and we've got more females than males also. Well, perhaps we could have a bias sample at this point. So, trusting the findings on this, we might not be able to do. And in experiment 1, we've got 32 high delayers, and we've got 27 low delayers. So again, we've got more high delayers than low delayers. So that could have an impact, but it's not too many. It's within kind of like a couple, so maybe it won't make a lot of difference. And then we've got 27 of the 59 who participated in experiment 1 agreed to take part in the functional neuroimaging study. So in the fMRI scan, as you can see on this picture here, that's showing like the different colours of it on the computer. So essentially, we've started off with 562 4-year-olds, of which 155 completed self-control skills in the 20s, then it dropped down to 135 in the 30s, and then 117 of them agreed to take part when they were 40, and then 59 of them agreed to take part. Oh, I've done that wrong. So let's start again. 562 4-year-old children at the nursery did the marshmallows task. Then, when they were 20, only 155 of them completed the self-control scales. When they were 30, only 135 of those completed the self-control scales. And then of those 135 that were left, 117 of them were contacted because we were looking for those above or below average, and only 59 of them wanted to participate. And then of them, only 27 of them participated in experiment 2. And in that, we've got 15 high delayers and 11 low delayers. And we've got one 41-year-old man also being excluded from the sample because he had poor results. So he would have been kind of like an outlier and would have probably impacted on the results. We kind of don't include outliers because they might distort the mean. If we use a mean value, obviously it might be better not to use a mean, then we would have to kind of take that one participant out. It's not great really that they've taken one out because there could be something about him. Maybe he's more representative of everybody else. We don't really know. So obviously we're going to take it out in terms of statistical reasons because it'll distort our mean value. But we really shouldn't be excluding people because there could be something about that that explains biases in the sample. So the procedure for experiment 1, this tested whether individuals who were less able to delay gratification as children, so the low delayers, would also in the 40s show less impulse control when they had to suppress their response to a hot relative to a cool cue. So hot cue being that kind of like scary face or laughing face or whatever relative to a neutral face that's cool. So 59 participants consented to take part, as we mentioned before. These here would be kind of cool faces. The neutral, they're not kind of showing anything. So participants completed 2 versions. The cool version, which is this one here, they've got male and female stimuli. So, being shown faces of males and females. One sex was go, which is the target. So they might say, every time we see a female face, press the button. So that would be a go. Every time they see a male face, that's a no-go. So they shouldn't be pressing that button. So that's the non-target. So basically, when it comes up, if it's a female face, they will go, that's fine. But when the male face comes up, they've got to suppress that reaction so that they don't press the button because they're not supposed to be going on that. So before the onset of each run, they've got a screen. It indicate which stimulus category serves as the target, so when to press the button, is it male or is it female? And they were instructed to respond as quickly and as accurately as possible. So quickly because, we want to make sure that they're not overthinking it and it's their immediate response, and accurately because we want to make sure that they're getting it right so that we can see whether or not they are suppressing their responses to those cues. Each face is appearing for 500 milliseconds. So it's very, very, very quick. So that's like half a second. Followed, or is it a fifth of a second? Let's just see. Yeah, I googled it. it's half a second, followed by a 1 second interstimulus interval. So they flash the face for half a second, they've got to respond to it, and then they've got a second gap, and then another face will come up. So it's a very, very quick procedure that they've got to, you know, do. They've done a total of 160 trials per run in a pseudo randomized order. So 120 go and 40 no go. So basically, there's 160 faces popping up in a run. So that's, you know, it seems like a lot, but if you think about it, you're getting like a face for half a second and then a second interval. So basically, every face is like 1.5 seconds. It's not a lot of time, so you'll get through 160, you know, pretty quick. And they've got more goes than they've got no goes. Because if you think about it, if you're going quite a lot, you'll get used to going, and then when the no go pops up, it's more difficult to then suppress it. So that's been used as a kind of control. And this pseudo randomized order means that it's kind of like, you know, on countdown when they press the numbers and it comes up with one. So it's not an actual, it's not random because it's not chance. It's kind of based on kind of an algorithm that the computer uses to kind of come up with the number. But this means that it's not going to be affected by things like order effects because they're all in a different order. So it's not like some faces will be more difficult than others and that kind of thing because they're all in a different order for everybody. So it should cancel out any problems like that between participants. The task was a 2x2 factorial design. So what that means is, you know, if you were to draw a summary table, it would look like this. You've got male and go as a result. You've got male and no go. You've got female go and you've got female no go. So you'll have a score each of those in terms of reaction time, in terms of accuracy. So you've got kind of 4 results from it. So it's kind of a 2x2 because you 2 times 2 is 4. You get 4 results from it. Factorial design is kind of important when you're doing statistical tests, which we will learn more about later on. In particular, parametric tests, which I'll talk about in a minute when we go through the results. We learn about non-parametric tests. If you do psychology A level, you'd learn about parametric tests. So we will briefly go through some of it so that you kind of understand it a bit better, but it's just to know that it's a 2x2 factorial design. So we've got accuracy and response time has been calculated in 4 runs representing each combination of stimulus sex. So you're going to have go on a male, go on a female for hot cues, and you're going to have go on a male and then go on a female for cool cues. So you're going to end up doing it 4 times basically. And the hot version is identical to the cool version, except that you've got fearful and you've got happy facial expressions. As I mentioned before, these are processed faster by our brains because, evolutionary speaking, we had to do it because we needed to survive. If people are looking at us like that, it's telling us something about what's in the environment. So we need to know and be able to interpret that to keep us safe. So we have to process those faster, which means that those alluring cues become very, very difficult to suppress and to try and, you know, not go on those trials because of how those faces are being processed by our brains. Now, these tasks have been presented using programmed laptop computers that are sent to the participants' home. So the participants are doing these go no-go tasks at home. Now you might say that's great because it's ecologically valid, they're going to feel more comfortable, probably going to show less demand characteristics because they don't feel like they're in an artificial setting, so it's not going to affect them as much. But then you might say, you know, they could be distracted by things in their environment, so that could affect, you know, their ability to do it. You know, maybe they've got a child and the child started screaming in the middle of one of the trials, then, you know, that's going to maybe affect it. I'm not saying that did happen, but we don't know for sure, you know, what was actually happening in those participants' homes. But then it is on laptop, it's quite reliable, it's standardized, you know, everyone's getting the same task, so they should get the same result of, you know, at the end of it in terms of no-go result, unless of course they've got extraneous variables distracting them. That could create an unreliable result. Experiment 2, we've got an fMRI scanner that's used, as we mentioned, to examine those neurocorrelates, so whereabouts in the brain is being activated. And these scans here show activation of the ventral striatum. It was anticipated that for low delayers they would show less activity in the right prefrontal cortex, where the inferior frontal gyrus is, and amplified, so greater activity in the ventral striatum compared to high delayers. So, low delayers want that immediate reward. They've got lots of activation in the basal ganglia reward system where the ventral striatum is. They want that reward. Whereas high delayers have got more activation in the inferior frontal gyrus because that's the one that's trying to control and suppress what's coming from the ventral striatum. So there should be more activity there. The 27, as we mentioned, participants agreed, consented to complete this, and they completed a hot version of the go no go task similar to the one used in experiment 1. There was differences in timing, number of chart trials, and the apparatus that were used, because obviously they're in a scanner, so they've got to use kind of different apparatus in terms of that. So they've just used that hot version task. This is what they would have had to have looked like in the scanner. So they're doing this task whilst they're like that. So you might say, they're probably really uncomfortable, probably not going to be able to do it as well as if they were just sat at home on a laptop. So maybe we can't really compare those findings against each other, validly. Each face in this one is presented for the same amount of time, so 500 milliseconds, but they've got a jittered intertrial interval, which ranges between 2 and 14.5 seconds, with the mean being 5.2 seconds. So they don't really know how long that interval is going to be. It could be anywhere between 2 and 14.5 seconds. They're doing a difference of 48 trials, so they're not doing the 160 anymore, they're doing the 48 per run. And again, it's in a pseudo randomized order, but this time we've got 35 go and 13 non-go. So there's still a lot more go than non-go. In total, imaging data was only acquired for 26, because remember they've excluded one of them because he had very poor results. And 70 go trials for each expression. The task was viewable by a rear projector screen. So as they're in the scanner, as you can see on the top picture, they've got a screen like this, and they're using a keypad that is on the bottom. This is called a NeuroScreen 5-button response pad, and it basically records their responses and their reaction times. And as I mentioned, one of them has been excluded because they had poor performance. Now, for this experiment, we've got a 2x2x2 linear mixed effects model. So this one's, way more complicated, because you've got, within subjects means repeated measures. So they're doing both the go and the no-go task. You've got the emotion, happy and fearful. Again, they're doing both of those. That's within subjects, repeated measures. And whether a high or low delayer is between subjects, which means independent measures, they're only taking part in one condition of the IV. You can only be a high delayer or a low delayer. You can't be both. So this is kind of what the table would look like. You're going to have a score for the high layers for if it's a happy face, on go and no go, and if it's a sad face, on go and no go. And then same for the low layers, you're going to get a score if they're happy and they go, if it's happy faces and it's no go, if it's sad faces and it's go, and if it's sad faces and it's no go. Again, it's the same kind of sex, go and no go. So go on male, don't go on male. Instead go on female, that kind of thing. But they'll be seeing happy faces, they'll be seeing sad faces. So they're the hot cues that they're seeing. And 2x2x2 is 2 cubed, which is 8. And as you can see, if you count the boxes, there's 8 different boxes, there's 8 different results. So that's why we say it's a 2x2x2, because at the end of it, we've got 3 conditions, and we've got 8 results from the end of it. Now, cases use what's called a parametric test, which is ANOVA. And when you calculate ANOVAs, if you watch this video, it will explain how ANOVAs are done. Now, it's a long way of doing it. If you go to university to do psychology, they have a statistical package called SPSS, and it does it for you. You just need to understand kind of what an F value is and how you interpret that and know what that means. So basically, they found that reaction times outside the scanner in experiment 1. So they're not in a scanner in experiment 1 anyway. So I don't really know why that's on there. It's just reaction times. There's no effects of delay group gratification on reaction time measures to correct go trials. So basically, they can go on a go trial fine. There's no, delayed gratification doesn't affect their ability to go. That wasn't an issue. And it says that these findings, P is greater than 0.1, and P is greater than or equal to 0.9. So really they're not significant anyway, so maybe we shouldn't even accept those values because P should be less than or equal to 0.05 really. So this is saying that actually the probability is greater than 10%, so these results could even be due to chance, so they're not great anyway. Experiment 1, when you're looking at kind of an ANOVA, you compare it against what's called a critical value. If you've watched that video, it will make sense, and you can use this table for it. So if we're looking at the significance level of 0.05, that's suggesting that anything we've found is less than a 5% chance as being due to chance factors and extraneous variables. So it's 95% sure that what we found is true. You would use this table to kind of look it up. So have a look at the video and it will explain how to use it. We've also got a video here to explain p-values in more detail, so please watch that also. You don't need to know what z-scores, alphas are, but p-values and critical values it will explain in detail for you. Now, accuracy outside the scanner, so how accurate the person was in that experiment 1, so looking at the cognitive go, no-go tasks. They had a high level of accuracy for responding to the go trials during both the cool and the hot tasks, so they're pretty much correct. We've got a margin of error because obviously we've got human error in everything. And the low and high delays performed with comparable accuracy. So in go trials, we've not got a problem. They're fine. If you tell them to go on a go trial, that's fine. There's no kind of interference between the ventral striatum and the inferior frontal gyrus because the ventral striatum wants to go and the inferior frontal gyrus wants to go also, because that's what the aim of the task is. So they're both in agreement. So we've got no kind of conflict, no suppression needed. It's fine. Now, the accuracy for the no-go trials was more variable. Now, low delays committed more false alarms than high delays. Now, a false alarm means they've pressed the button when they're not supposed to. So if the task was go for a female, then they might have pressed the button on a male. So they couldn't suppress that want from the ventral striatum to go. Now, low and high delays performed comparably on the cool tasks. So the cool tasks, the kind of, didn't affect them so much, but it was on the hot tasks that they struggled. So they found that more difficult to suppress what they wanted. So what the ventral striatum wanted as the reward. So overall, they produce differences between high and low delayers. If you've got emotional hot cues, then low delayers are going to find that a lot more difficult to suppress than they'll need to go on that trial. So they can't really suppress the response to happy and fearful faces. They find it a lot more difficult than higher delayers. So there's some suggestion there that there's the inferior frontal gyrus is more in control with those high delayers. But we don't know that yet from experiment 1, because remember, experiment 1 is just about cognitive. It's only really in experiment 2 when we fMRI scan the brains that we find out that it's these kind of brain areas that are responsible. Now, for experiment 2, they've used a paired samples t-test, again another parametric test. If you watch this video, it will explain what that means. And for the reaction time inside the scanner, we've got the two delay groups did not differ significantly in reaction time in go trial. So again, exactly the same thing as before. They're not really differing in terms of reaction times for the go trials. It's not a problem. But for the hot trials, it's a bit different. Well, accuracy for hot go is fine, but it's accuracy for no go if it's hot. So the false alarm rate is comparably different. They basically agreed with experiment 1. Low delayers found it a lot more difficult to suppress the reaction to those cues. I mean, they're doing the same task, they're going to find the same things in terms of accuracy and reaction times inside and outside the scanner. So, I mean, that's showing some kind of agreement there that even though they're in a scanner, they're still performing the same way. So maybe that provides us with a bit more kind of support for what we believe when we're inside the scanner in terms of what brain regions are activated. So they've found that there are different areas involved in the go and no go trials. We've got the right inferior frontal gyrus is involved in accurately withholding the response. That is in control of whether that person goes or no goes. High delays found it a lot easier to suppress those hot cues, those alluring cues, than the low delayers did. There wasn't as much activity for the low delayers in that area, so it suggests that it's not as good, it's not as developed in terms of brain region as it is for the high delayers. And the ventral striatum has demonstrated significant differences. The reward-related region is showing a kind of three-way interaction between the group, the trial, and the emotion. They've got more activity in the happy no-go trials for low delayers relative to high delayers. So it's kind of showing that when you've got that alluring cue, the ventral striatum's got more activation for low delayers than it has for high delayers. So in conclusion, we've got a sensitivity to environmental hot cues. If we see somebody, smiling face or fearful face or angry face, it's an alluring cue, and it plays a role in whether or not we can suppress our actions towards that. Do we, we might want to respond in kind, so in the same way, but it might not be appropriate, so we kind of have to suppress, and that's what our inferior frontal gyrus is for. This resistance to temptation to respond to that alluring cue, measured by that delay of gratification task when they were 4 with the marshmallow, has been found to be, a prediction of what they're going to be like when they're older on cognitive go and no-go tasks. So it's it's suggesting it's a stable individual difference. This delay of gratification is stable throughout your life. It predicts, a reliable kind of prediction of the circuitries that are being used in your brain and how you're motivated to control that process. It suggests that your capacity to resist this temptation varies by context. The more tempting something is, the more likely that person needs to regulate their behaviour. So if they can't do that with the inferior frontal gyrus, then they're going to be a low delayer. If they can, they're going to be a high delayer. It's suggesting as well that it's not just a behavioural explanation, that you've also got brain regions that are involved in it. This is the reference to the study. You have to be able to write this in full and know where all the commas and the full stops go. We've got kind of all the researchers in the beginning. It's Casey et al. Because it means Casey and others, so there's lots of different researchers. It was done in 2011. The title of it is Behavioural and Neural Correlates of Delayed Gratification 40 Years Later. The journal that it comes from, so the big book that it comes from, is the Proceedings of the National Academy of Sciences. It's volume 108, number 36, and the page numbers are huge. Look at those, 14,998 and 15,003. That must be a blooming big book if it's that page, unless it's just an electronic version. I don't know, but that's huge, isn't it? Okay, evaluation of the study. Now, it's great. We've got quantitative data. We've got lots of statistical tests. Suggests it's going to be, more valid. It's very easy to compare between the different, high delayer and low delayer, go and no go, hot cues and cold cues, different brain regions. So it's very easy to compare and analyse between those if you've got the software, obviously, to do it and collect the data in that way, which Casey has. So it's more objective. There's no kind of research bias and interpretation of results. It's just there. It is as it is. So we can trust the results in that sense. We've got a naturally occurring IV of a quasi experiment. You can't do it any other way because you're either a high delayer or a low delayer. There's no kind of you can't force someone to be a high delayer or a low delayer. This can be quite socially sensitive. If somebody is finding out that they're a low delayer, and then they're not going to be as successful, you're kind of giving them a label, which is very unethical, and they might have a self-fulfilling prophecy. They might believe the label and then just become a low delayer anyway. It's very, very difficult to suggest that, okay, this research is suggesting it's stable, but maybe because when they were 4 they were told that they were a high or low delayer and it's impacted them in their life. Who knows? it might not be stable. It might, be reactive to things in the environment and stuff like that. This is very kind of reductionist. It's just looking at that kind of nature, that biological explanation for behaviour. It's not kind of considering, the environmental, the educational impacts. They're all at the same nursery. Maybe that's had some impact. And then where have they gone afterwards? There's no kind of information about that. Um, you might say as well that we've got them all from one nursery. And Stanford, big university nursery, um, the people at Stanford University might be quite affluent. They might be a specific kind of person. Maybe the children are actually all quite high in terms of delayed gratification, in comparison to what might be found in the general population. So maybe we can't apply these findings to everybody. Maybe it's only applicable to certain groups of people. It can be quite ethnocentric. Although, studies have been replicated and found similar across different countries, but this study itself doesn't look at that. So this study itself is ethnocentric. Um, it's got mundane realism in the sense that, it's laptops in their own environment, which could be a task that they normally do. Although, you don't really get in a scanner and do a go no go task. I mean, you don't do a go no go task anyway, do you? So maybe that's not something you would normally do. The marshmallow one's probably got a bit more mundane realism because, that probably would happen, if you were a child and you were with your parents or something. So maybe that has got a little bit. The repeated measures in terms of the self-control scales when they were 20 and 30, maybe there's going to be some order effects there, some practice effects. Maybe they're showing demand characteristics because they know what it's about. They want to come across as a highlight to be, socially desirable. There's lots of high control and standardization. Everything's the same for everybody. They've all got the same laptop, same go and no go task. It's pseudo randomized. All that stuff's kind of good. It's very scientific. So this study's got lots of good things about it, but then it's got lots of limitations because it can't really explain everybody's ability to delay gratification and whether it's stable across time and whether the same brain regions are implicated in everybody. So whilst it is good and illuminative in explaining delayed gratification in terms of cognitively on the behavioural level, and also biologically in terms of its neural correlates, we've got to be very careful in how we trust these results, because they could be socially sensitive and have impacts for people in the long term, and that's not ethical.